And for the discussion, once more again. And we go to the second presentation on the uptake of nanoparticles into cells and skin by Eckart Rühl from Berlin, Physical Chemistry, FU Berlin.
The priority program on bio-nano interactions. And in addition, I would also, whoops, here's something wrong. Doesn't like this. Okay, I'd also like to acknowledge the contributions of other collaborators who contributed to the work I'm going to show. First of all, this is a brief outline. I would like to start with synthesis because nanoscopic objects always start with synthesis. Without the materials on hand, you cannot do any experiments. Then I will talk briefly about the characterization of these particles. I will also address similar issues than were just addressed in the previous presentation. And then I will talk about uptake studies into cells and skin. And finally, I will sum up. The particles that have been used in the priority program are mostly inorganic particles. They are either silica particles or iron oxide particles, where core shell architectures play a crucial role, because these allow us to substructure the particles to include, for example, molecular probes or possibilities for distinct probing of the nanoparticles, and also the variation of the surface material allows us to covalently bind functionalizations that are of importance for the interaction with the biological media. This is a more complicated architecture of particles that we use, because in the middle there is a gold core. This is, as a high Z material, advantageous for X-ray absorption. And then we also can bind either quantum dots or dye layers, and then we can surface coat the entire system as we like. So the internal structure is quite important. For the diagnostics, we can also vary the shape of the particle. This is just an example of rod-like particles, because the question is always, when there is interaction of biological matter, whether the size and shape matters. And finally, we learned that the functionalization of the particles is of crucial role, because this is what interacts with biological media. So the neat particles, they can be compared to a whole variety of different ligand systems. We'll not go through all of them, but you see here that the surface charge that leads to electrostatic stabilization of the particle, so that there is no aggregation in a solution or cell media, can be well adjusted by the zeta potential. And I will just go here through a few functionalizations of positive zeta potential, because we learned that this is advantageous for cellular uptake because of the negatively charged patches on the cell membrane. And I'll just show here the APS ligand, the H-APS ligand, and the PEG-silane ligand. These are ligand systems that we studied regarding their uptake behavior more intensively. And just to give you a flavor how the different ligands, they have all positive zeta potential, these amine functionalized particles, they tend to be either stable in water or in cell media, or they tend to aggregate. And what one wants is, of course, that these particles stay isolated, so electrostatically stabilized, so that one wants to have no increase in diameter as a function of time. This is here the ratio of diameter, that is the ratio of the actual diameter divided by the initial diameter probed by dynamical light scattering. We want that this should remain at unity. You see here at the AHAPS, this gives fairly stable conditions, whereas at the APS ligands, this is highly unstable. So after a short time, a few hours, these particles start to aggregate. They follow wonderful exponential aggregation behavior. And this is a mechanism where this ligand pulls itself off the surface and leads to a loss in stability and massive aggregation. That this is not just an artifact of dynamical light scattering can be seen independently by transmission electron microscopy, where you see the neat silica particles, they're more or less isolated, even more isolated the HAPS, the stable ones, but then the PEC silane functionalized ones and the APS one, they form aggregates. And this is, of course, of importance. And a nanoparticle that changes its size just by aggregation has the same properties as a system that stays isolated. So for nanoscopic objects that undergo uptake, one really wants to have such isolated particles that do not aggregate. The other issue that has been discussed before is what happens to the particle surface that is properly functionalized regarding the absorption of proteins. And this is an experiment we did on iron oxide particles that have the beauty that you can keep them in a magnetic field, and then you can expose them, for example, to a blood plasma, and you can flush them and look what kind of proteins come off, turning on and off the magnetic field, and look at the different properties. This is the quantity of adsorbed proteins as a function of dilution of the plasma. So this simply shows that this scales with the quantity of plasma that is offered. If one does gel chromatography, one can look at the speciation of the particles, so the little albumins, then the major fraction of the immunoglobulins and the fibrinogen and a few others. And as a last item, we looked at the function of incubation time, whether there is a change in protein corona. So this issue of the Roman effect, whether the protein corona is stable or not. And these results clearly indicate that as a function of time, there is no change in protein corona, so that these proteins stay when they are adsorbed. So there is no evidence for a Roman effect. The next step is, once we know about the particles, their physicochemical properties, their protein absorption behavior, what are their uptake mechanisms into cells? So we look at the endocytosis pathways, and here you see spinning disk fluorescence microscopy images on these different silica particles, either neat particles where we see very little uptake. This would show up as green dots here. This is the APS. You only see outside on the cell membrane big patches. So this is just these aggregating particles. They stick on the surface of the cells. These were HeLa cells. And finally, these APS functionalized particles that do not aggregate. Here you see that there is uptake. You can see this in colocalization studies, and further experiments indicated that they end up in lysosomes or late endosomes. Once we know about the cellular uptake processes, we looked at skin, mostly human skin, where it is of importance what are the uptake routes, because the skin is well shielded to any hazards by the top layer, the horny layer, the stratum corneum, and there's some weak part. This may be the hair follicles. So the dermatologists, they discuss whether this is an entry route of either nanoparticles, so unintended uptake of particles, or for vaccination, a possibility, or whether sweat glands or inter or intracellular transport processes through the stratum corneum into the epidermis, into the dermis may occur. The optical microscopy or fluorescence microscopy clearly shows that there is uptake into the hair follicles. You see here these stained areas. These are stained nanoparticles. And also the different skin cells, the Langerhans cells and the keratinocytes, they have different uptake behavior. So there is stronger uptake by the Langerhans cells, as you can see here. The colleagues from dermatology, or all those who have just access to fluorescence microscopy, are wondering whether this image that you get from fluorescence microscopy, this is just the stratum corneum where all fluorescent particles are sitting, whether this is correct or not, or whether some particles can get deeper. But of course, if you use fluorescence microscopy, it is very difficult to resolve individual nanoparticles. And therefore, we try to look with different techniques. I'm sorry for this. By X-ray microscopy, a way to probe with chemical speciation nanoparticles with high spatial resolution that can be as low as 10 to 20 nanometers. This requires synchrotron radiation, where the radiation is focused onto the sample by a Fresnel zone plate and then imaged on a detector, or as a variant, the scanning X-ray microscopes may be used, where the sample is scanned just by a piezo scanner. The important part is that the tunability of synchrotron radiation gives rise to chemical specificity due to molecular transitions, so that we know exactly what is excited. This is just for showing with high resolution that individual particles, these are these particles with a gold core and the dye around, so that we can use the same sample for both approaches, that they occur mostly here on the top layer of the stratum corneum, and some particles get a bit deeper, but everything is in the topmost horny layer of the skin. So we see here that these particles do not aggregate, but they stay on the skin surface. We also looked at hair follicles where the particles get in. This is here the contrast from the hair follicle opening. So between the hair and the wall of the follicle, here you see the particles. So this is nothing where any uptake into the Langerhans cells and so on is observed. Even more exciting is the possibility that one can get three-dimensional information from X-ray microscopy. This technique has the advantage that entire cells can be investigated, because the samples can be much thicker than in electron microscopy, and also wet samples can be investigated. And we're wondering in keratinocytes whether there is uptake of the particles into the nucleus or not. So the idea is just to tilt the sample and to take images. You see this here, hopefully. This is a series of microscopy images, but it is much better not to just to tilt the sample, but to construct from this slices, where you see then step by step that the membrane of the nucleus shows up, and we're interested to see whether here's any uptake, so that we're not fooled by projections. And it turns out that for these large particles, there is no evidence for any uptake of the silica particles. The only exception for uptake in the nucleus was found by gold particles of roughly 50 to 60 nanometers. Here you see the projection. These are inside, and these sit on the membrane outside the nucleus. And this can be shown here. Here we go. That you will see here, this is the evidence that some particles are evidently within the nucleus and the others remain outside. So this gives us insights into three-dimensional information of nanoparticles. The other question, of course, that is with skin of importance, what happens if the barrier is damaged? And this can be due to diseases, this can be due to mechanical changes in the stratum corneum, so that the particles get deeper through this layer into the epidermis or even later. So our colleagues from veterinarian medicine, they changed the barrier by tape stripping and by oxazolone-induced allergic reactions. And they looked for a variety of different days for changes in the uptake behavior. And to take this story quite short, is there was no uptake of these particles into deep skin layers. They all stay outside here in murine skin, so that the damage by tape stripping, just to rip off the upper layer of the stratum corneum, or by this kind of allergic reaction happens to lead to uptake. There's one exception, and this is mechanical damage. This is here a pricked skin, where just the skin is mechanically damaged. You see here such a area where the skin is pricked. Outside sit the majority of particles, but if one looks carefully, there's also another mechanical damage. Here, several tens of microns further inside there, one finds particles that are uptaken, and if one looks in close-up, they're here, and these dendritic Langerhans cells, there's uptake of these particles, so that once mechanically the barrier function is lost, there seems to be uptake into deeper skin layers, so near the stratum basale where these particles were found. This brings me to the last item, the intentionally induced drug delivery. So one can use the knowledge from the inorganic particles to derive drug delivery approaches. For example, here by nanocarrier, these can be dendritic systems, this can be other polymeric systems that can be triggered in drug release in order to see whether in inflammatory skin diseases there is a way to deliver the drugs in a more rational way where the drugs are needed, and especially for those drugs that are difficult to formulate, so hydrophobous drugs, drugs with a higher molecular weight that are wrapped into these polymeric systems, and then to probe either with labels or without labels, where is a drug and is it really transported to the site of action. I just show you here as one example the delivery of dexamethasone, a standard anti-inflammatory drug that has been put to human skin. And just to stay with the topic of X-ray microscopy, this is X-ray absorption of fixated human skin, and dexamethasone This is excitation at the oxygen KH, and you can easily distinguish, as you see here, from the resonance of the oxygen 1s to the pi star level, that the dexamethasone can be easily distinguished from the skin. We can even distinguish different oxygen sites. They have different absorption energies. So if you sit on this red oxygen here in the dexamethasone, then you have no interference with the skin, and you get pure chemical contrast. And this is shown here. This is an optical microscopy image. This is the stratum corneum. Here, this is the epidermis. Then comes the dermis. And this is the drug concentration. So you see most of the drug sticks in the stratum corneum. And if we integrate here, you even find down to the basement membrane an increased concentration of the dexamethasone. So we can probe with high spatial resolution and chemical selectivity in an absolute way, so either normalized to the square of the skin or to the volume, the quantity of drug, and we can also look at the impact of nanocarriers. Nanocarriers seem to increase the drug concentration in deeper skin layers, but it seems that only here in the epidermis, this here is the basement membrane at this cut, seems to be a much higher concentration. The other beauty is we can compare or cellular details from optical microscopy with X-ray microscopy. This is just an overlay, and if you look in detail, you see here that these structures, they're anti-correlated with the drug uptake, and we're wondering what that is. And if one compares this also with atomic force microscopy images, one finds that these are the corneocytes that seem to stick out. They cannot be well cut by the diamond knife. And this is exactly where the drug flows around. So in this brick and mortar model, the drug, the lipophilic drug, goes around these corneocytes and flows into the epidermis. And with high resolution, this is approaching the ultimate resolution. You see really the drug concentration in the individual layers of the stratum corneum, and also further inside in the epidermis and dermis, so that one can spatially resolve with of the order of 50 nanometers, one can look at chemical distribution of a drug within skin. So let me summarize with this. I showed you that we can prepare the nanoparticles. I showed you how the uptake works of nanoparticles into cells by colocalization studies, and I showed you a lot about X-ray microscopy as a way to, with chemical selectivity, to have high spatial resolution. And the barrier of the skin seems to be quite strong. So even if we have a tape stripping, that seems that the inorganic particles are not uptaken. The only exception are these polymeric nanocarriers. They seem to deliver more drug into the deeper skin layers. And with this, I thank you for your attention.
Thank you very much. Professor Schlapper apologized that he had to leave earlier, so I'm taking over leading the discussion. Any questions, please? Everything is clear? No questions? Beer, please.
Peter Gare, University of Bern. I wonder whether you did some quantitative studies. How many particles are actually going deeper into the skin, or is 100% outside?
That's an interesting one, yeah.
For the inorganic particles, it is very clear that almost everything stays outside. Only in the X-ray microscopy where this is in the per mil range, almost nothing goes in. And this fits with the general belief of dermatologists, but I think nobody has really quantified that due to lack of tools to do that.
What's the size of those particles?
Well, typically, these inorganic particles, they range between a few tens of nanometers to a few hundreds of nanometers. So this is the size range we looked in the inorganic, and the nanocarriers, they have sizes of a few nanometers, and if they're aggregated, they get to a few tens of nanometers.
Yes, please. Would you mention your name as well?
You showed two pictures that there were particles modified with APS and HAPS.
Yeah.
And I was wondering, what was the time of incubation with those nanoparticles? Because one of them were outside the cell and the other were inside.
Yeah. Typically, the exposure of cells, there are different time steps made. They range between 1 hour and 24 hours. This is the typical exposure times.
Okay, and did you check what's the shortest time that they can get in?
So for the uptake here, these into the nucleus, they were, as far as I remember, 6 hours.
Okay, and the other question, did you wonder what's the process of the uptake of those nanoparticles?
Yes.
Is that endocytosis or?
Yes, we know this. We did inhibition experiments, so it depends very much on the particle. They're either clathrin mediated or they're caveolin mediated.
Okay.
It depends on the material. So the iron oxide, this was caveolin mediated endocytosis, and the silica was clathrin mediated, and this was done by using dinosaur as an inhibitor.
Okay, thank you very much.
Last question, please, if that we have time. Maybe I have a comment or a question. The perception that the healthy skin is pretty robust against nanoparticles, is that still true?
Yes. Yes.
So, we don't really need to worry very much in terms of...
Except what I said with the only limit, it's mechanical damage.
Yes, okay,
yes.
That's understood. Inflammation as well, and sunburn?
This needs to be clarified, so this oxazolone allergic reaction, this didn't show anything, but it will be of interest for the drug delivery, whether when the barrier is weakened due to the atopical dermatitis or psoriasis, whether this is also true or not, we shall find out.
Is there any difference between light skin like ours and black skin?
We have to find out. So
this is black pigs.
This will be This will be a challenge because we rely on the samples from the plastical surgeons who deliver us with skin. And most of the skin samples we are from Caucasians, and we never had from black people any skin sample.
Yes, I understand. So thank you very much again for this presentation. Thank you.